in this particular module, we will study the different scope of microbiology. Now, scope of microbiology will include the microbe as such, their structure, their reproductive pattern, and how they are important to us. At the same time, what role do they play in nature? Microbes are universal in nature. They are present in the atmosphere, in the lithosphere, in the hydrosphere. And sometimes they do play very important role in recycling of different nutrients. And these cycles, as you know, are the biogeochemical cycle. And these cycles can be oxygen cycle, carbon cycle, nitrogen cycle. In the nitrogen cycle, which is essentially conversion of atmospheric nitrogen to soil nitrate, and this soil nitrate being reconverted to atmospheric nitrogen to maintain the high concentration of atmospheric nitrogen around 78%. Three major activities are observed here. They are the nitrification, which is conversion of nitrogen to nitrate by different microbes or the nitrifying bacteria. Then nitrate is reconverted to atmospheric nitrogen by the denitrifying bacteria. And at the same time, there may be the nitrogen fixers, which fix atmospheric nitrogen and enrich the soil with nitrate. Okay. Coming to another important cycle, which is the oxygen cycle. And this actually is including two major activities, that is the photosynthesis and respiration. Photosynthesis involves fixation of atmospheric carbon dioxide and utilizing it in the process of preparing food, which is carried out by photosynthetic bacteria and plants. And in the process, they release oxygen, which is returned back to nature, and in this way, the atmospheric oxygen concentration is maintained. But there is another type of cycle, which is commonly known as the sedimentary cycle. Now, why it is so called? Because the compound or the element, they are present essentially in the sediment. It can be the sulfur cycle or the phosphorus cycle. As you can see in this slide, in the sulfur cycle, there is conversion of sulfide to sulfate and reconversion of sulfate back to sulfide. And this is essentially carried out by different bacteria, that is the sulfur oxidation and sulfur reduction. The sulfur oxidation is carried out by the photosynthetic bacteria, the Chromatium and the Chlorobium, which is converting sulfide to thiosulfate. And at the same time, there is the thiobacillus, which is converting the thiosulfate to sulfate. So this process is oxidation of sulfur to sulfur dioxide or sulfate. And sometimes this particular sulfate is reconverted to sulfide by another group, which is sulfur reduction. And this is carried out by the anaerobes like the desulfo-vibrio, and thereby the level of sulfate and sulfide is maintained in the sediment, and it is called the sedimentary cycle. But apart from this useful activity, there are some pathogenic microbes, and they are causing innumerable number of diseases, because pathogenic microbes can directly enter our respiratory tract or alimentary canal, causing the disease. You consider the disease of plague, which actually removed lot of people. They died because of plague in the medieval Europe. Consider the disease of cholera, which is killing large number of people. But last 10-15 years, the outbreak of cholera was checked because of improved sanitation and other measures taken. At the same time, there may be some pathogenic fungi which can also cause dreadful diseases in human, which aggravates with the infection of HIV. Microbiology is a huge domain which involves the microbial structure, that is the diverse forms of microbes and their role they play in nature, that is in maintaining the different natural cycle, and at the same time the different compounds or secondary metabolite they produce, which may be economically important to human society. So all these are included in the scope of microbiology. But in this first lecture, we will try to see that what are the different microbes, what are the different structures, function, and their different importance. And then only we will get into their role in nature and also the industrial importance of microbes. Virology is the study of viruses. The virus particle, as it is called, is only formed of an inner nucleic acid and an outer protein coat. And on the basis of the shape of this protein coat, they may be spherical, They may be geometrical or sometimes binal structure. Upper part is geometrical and the lower part is helical, like what we see in bacteriophage. Viruses can also be classified on the basis of their host, that they can be plant virus, they can be animal virus, they can be bacterial virus, and on the basis of the nucleic acid, viruses may have the double-stranded or single-stranded DNA and similarly a double-stranded or single-stranded RNA. On the basis of the International Committee on Taxonomy of Viruses 2005, there are approximately about 2000 species, about 287 genera, 73 families and 3 orders recognized in viruses. And viruses are important not only because of the complete infectious particle or what is called virion, but also some associated structures which look like virus, but actually they are smaller than virus. And these particles may include the viroids. One such viroid is the potato spindle tuber virus or PSTV. As you can see in this slide, these are nothing but RNA molecules, and these RNA molecules cause the disease of potato spindle tuber disease, but it is only a part of the virus causing infection. Similarly, in the next slide you can see a very important structure which is actually a folded protein structure, and this protein is also known as a prion protein. And this prion protein is important because it causes a disease called bovine spongiform encephalopathy, or which is called mad cow disease. This prion protein is having two definite parts. One part is having the beta pleat structure, which is called the folded C-terminal domain, and the other part is N-terminal octa-repeat domain. And they, as you know, cause the disease of bovine spongiform encephalopathy, which is a neural disorder, can actually kill human, and it was a major problem in 1997, invading different parts of Europe. You can see the red marked areas, they are the actual areas where the bovine spongiform encephalopathy spread. Apart from that, viruses are very important structure which can control other organisms. And very strangely, this is a very recent discovery, there are certain virus which is called the Sputnik virus. And this Sputnik virus can actually take the help of the capsid of a satellite virus. So what we can see is that this particular Mimi virus can actually be degraded by the Sputnik virus. So thereby we say one virus particle is being controlled by the other. So this is what is called the virophage. Now viruses can also play a very important role in cancer. Viruses can alter the cell cycle and thereby they will induce tumor-like outgrowth. And one such virus you can see is the human papilloma virus. And this papilloma virus will cause the cervical cancer in female, and they will also produce typical wart in the skin. Bacteria can be classified on the basis of their structure, that is on the basis of their shape, which can be rod-shaped, round, comma-shaped, spirillum. And it can also be classified on the basis of their nourishment or nutrition. That can be photosynthetic or it can be heterotrophic, that is saprophytic or parasitic in nature. Photosynthetic bacteria can also be photolithotroph and photoorganotroph depending upon their carbon source. But most importantly, when we consider their structure, bacteria can be classified into two major types, that is gram-positive and gram-negative. That is on the basis of the gram stain that they retain or they lose. This is the gram-positive cell wall, this is the gram-negative cell wall. In the gram-positive cell wall, you can see this structure is the peptidoglycan structure. And this peptidoglycan structure is formed of the heteropolysaccharide of N-acetylmuramic acid and N-acetylglucosamine, which is interconnected by tetrapeptide linkage. Whenever they are stained with crystal violet with iodine, the stain is retained, so they are gram-positive. But when we come to gram-negative bacteria, you can see that there is an outer lipopolysaccharide layer followed by the peptidoglycan, which is only 10 to 15%. And there is an inner membrane. Of course, there is an intermediate area or the matrix. So what happens here, that because of the peptidoglycan area being very small, the gram stain cannot be retained. Bacteria similarly can also be classified on the basis of the flagella. They can be without flagella, that is atrichous. They can be with a single flagella or monotrichous, with a tuft of flagella or lophotrichous, with flagella on both the poles or amphitrachus and flagella surrounding the bacteria, that is peritrachus. And you can see in this slide the respective examples, that is monotrichus is Vibrio cholerae, then this is Bartonella, this is Spirillum, that is the amphitrachus, and the peritrachus is normally the E. coli. Vegetative reproduction is by the process of amitosis, meaning the nuclear, here it is not the nucleus, it is called the nucleoid, divides directly without the formation of any spindle fiber. And this division involves binary fission, that is one mother cell is dividing into two daughter cells directly, and it is performed very quickly and normally seen when there is adequate nourishment in the nature. Another way of reproduction is the endospore formation. Endospore is a structure which is normally exhibited when the environment is little bit difficult, when the nourishment is less or when the water is less. And this endospore formation normally involves formation of a three-layered structure, and this three-layered structure will have the protoplasm shrunk within. And after the endospore is formed, normally bulk of the cell is lost. The endospore may be terminal or intercalary in position. But the endospore usually is a device by which the bacteria sustain the unfavorable condition. And after the favorable condition is regained, this endospore will germinate to produce the normal bacterial cell again. Now, bacteria can also be studied with respect to the different bacteria-like or prokaryotic particles. And these particles may include the different forms of prokaryote. And they may be the mycoplasma, which is commonly the pleuropneumonia-like organism. Now, why it is called pleuro? Because they don't have any cell wall. You can see in this slide, they have a typical prokaryotic structure, but the outer membrane is a three-layered lipoprotein membrane. So they take different shape. Because of the changing nature, they are called pleomorphic. Similarly, they can also induce the infection of pneumonia, and so they are called pleuropneumonia-like organism. Coming to the next group, which is the Rickettsia. Rickettsia is also similarly a rod-like bacteria, but this Rickettsia also can invade human or other animals, and they also can cause pneumonia-like infection.
Sir, what can be the current status or the future positioning of Rickettsia?
Okay, you see, considering the cell structure, Rickettsia is considered under alpha proteobacteria. Their protein coat is similar to certain viruses, but because viruses do not have any cellular function, whereas Rickettsia have their independent cell division, it is better to consider Rickettsia under prokaryotes. Now, apart from Rickettsia, there are other organisms which also belong to bacteria-like group. They are the spirochetes, which can cause diseases like neurosyphilis or normal syphilis, and there may be other organisms like chlamydia, which is also intermediate between bacteria and virus, because they do not have proper cellular structure. They have the elementary particle. And the last one, that is in the prokaryotic group, is the actinomycetes, which are mostly the soil-borne organism, but they have a typical mycelial structure, and they also have spore form similar to fungi. So it is actually considered an intermediate between bacteria and fungi. Phycology, which is the study of algae. Blue-green algae, as you know, is cyanobacteria. And this cyanobacteria is now placed under the prokaryotic group. Considering the oldest algae, it was found out that the Precambrian era fossilized algae was recovered from the Vindhyas in India. And this group of algae represented photosynthetic algae. It was called prochloron. But from this particular unicellular green algae, different other forms have evolved, and these forms are the green algae, the yellow green algae, the brown algae, the red algae. We will start with the colonial green algae here, which is the volvox. The volvox is formed of a colony of cells where the outer cells are actually having flagella, so they help in locomotion. Whereas the inner cells, they are actually photosynthetic units, and they also have a storage starch in them. in their pyrenoids. At the same time, you cannot see that in this slide, but there are cytoplasmic connection. So there is a functional integrity between these cells. And the largest cells, what you can see here, is actually represented by daughter colonies, helping in reproduction. Coming to the next one, this is a filamentous algae, commonly represented by the yellow-green algae, because they have chlorophyll, at the same time they have carotenoid. You can see that there are two reproductive structures here of that particular algae called Vaucheria. There will be the Ugonium and there will be the Antheridium. And this slide actually depicts their sexual reproduction. Coming to the next group, which is commonly called the Bacillariophyceae or the diatoms. You can see some cells are round and some cells are elongated. The round cells are called the centric diatom and the elongated cells are called the pinnate diatom. But this pinnate diatom, normally, if you see it from the side, they look like a petri plate, having two valves, the epitheca and the hypotheca. So whenever they reproduce, the two valves separate. And when the two valves separate, they produce a structure called oxospore. These oxospore actually help them to maintain their size. They are formed of silica. So they are deposited as siliceous earth and they are commonly called diatomaceous earth, which is a very good refractory material. This is a higher group of algae represented by this brown filament, and this brown color of this algae is because of a brown pigment or golden brown pigment called fucoxanthine. But apart from fucoxanthine, they may also have chlorophyll pigments. The red algae represent the common seaweed and the red color is due to phycoerythrin. And this red algae may contain different stored compound which actually can be of different economic importance. One of them is agar-agar, which is a unique compound, a neutral starch, which is used in preparation of microbiological medium. Coming to the reproduction of algae, it can also reproduce by vegetative means. Vegetative means they will reproduce by fission or by fragmentation. That means if there is a filamentous algae, the filament will separate and then they will produce a new plant body. Apart from that, there may be asexual reproduction through zoospore and sexual reproduction, which I have already shown, that is by fertilization. Mycology, which is the study of fungi, a group of non-chlorophyllous organisms which can be unicellular or filamentous. Now, the earliest record of fungi is in the form of Prototaxitis, as you can see in this slide, which is having a lamellated structure with some definite green areas within. But this particular fungi is considered to be a fossilized fungi.
Sir, in some members of Prototaxitis family, there is formation of medullary spots. Can they be called a lichen?
See, these medullary spots are pigmented spot. So there is an option that there may be algal colony present within. But considering the broad nature of this fungi, which is mycelial in nature, we normally consider it as a fungal member, though there are some specific pigmented area which just represent some algal colony within. Now coming to Myxomycetes, this group of fungi is not having typical mycelial structure. You can see they are formed of a typical thalloid nature and they are producing sporangiospore. Fungi can also be classified on the basis of their reproductive spore. The first group is reproducing with zoospore, which are flagellated spore. The second group, which is normally called the Zygomycetes, because of this presence of the thick-walled zygospore, and this zygospore is a diploid spore which is act as a resting spore and will germinate to produce haploid mycelia. Another group is the Oomycetes, which is producing the oospore, and this is also a diploid spore but not having that thickened or echinulate outer wall. But all these fungi, they represent the primitive group because the mycelia is without any septum. But the higher fungi is represented by two major groups, the Ascomycotina and Basidiomycotina, having this ascus and ascospore and Basidia and Basidiospores. The difference being the ascospores are present within the ascus, whereas the basidiospore is present outside the basidia connected by sterigmata. Apart from them, there may be another group called Deuteromycetes or Deuteromycotina, where there is no sexual reproductive structure. Now, the life cycle pattern, there is alternation between haploid and diploid stages. But here you can see there is an intermediate stage which is dikaryon or N+N stage. That is, the nucleus will divide within the filament, and then one of the nucleus will ultimately pair with the existing nucleus and produce the N+N structure. And this stage will ultimately produce a diploid stage, or they continue to remain as N+N and then they separate. Now, fungi normally can be used for different economic importance, and these fungi can actually produce secondary metabolites like antibiotics. They can produce different organic acid, alcohol, and at the same time fungi has got a very important role in bioremediation. That is, they will remove toxic effluent and they will make the nature pure from this toxic effluent, including heavy metals. And at the same time they might produce some very important drugs like some anti-cancer drugs, which is produced by a higher bracket fungi, the Ganoderma. And this Ganoderma will actually be considered as a wood rotting fungi because it grows on wood, but yet it is so novel, so important for medicine industry.
Sir, I would like to know that since Ganoderma is so important, can this association with the bacteria affect its shelf life to any extent?
Yes, normally anytime the fungal fruit body is exposed to bacterial infection, so they also destroy the fungal fruit body and the shelf life is reduced. But if sometimes there is an algal consortium on the existing fruit body, this bacterial infection may be reduced and thereby the shelf life might be increased. So, fungi sometimes can also act as source of infection to plant, which is considered as plant pathology, and at the same time they can also be inducing infection in human, which is commonly called mycosis. And they are causing innumerable number of diseases of the skin and the internal organs, to which sometimes we don't have any answer. Protozoology, or the study of protozoa. It is considered under parasitology because majority of these protozoa, they are unicellular animals causing infection to human. And protozoa can be classified on the basis of their nature of locomotory organ. Sometimes they may locomote with the help of pseudopodia, as you can see in this particular cell, which is an amoeba. They are locomoting with the help of pseudopodia, and they are also followed by another group which is the plasmodium, and they are reproducing by unique structures called sporozoites. Similarly, there may be another group which is reproducing like the previous group, but they are locomoting with the help of cilia. So they are represented by giardia. Coming to their life cycle, now essentially they may be divided into two stages, the infective stage and the resting stage. The resting stage is represented by the cyst, and the infective stage normally is the trophozoite. Coming to their reproductive pattern, now most of the amoeba reproduce by binary fission, which is somewhat similar to amitosis, that is direct cell division, and these are the two cells which separate and produce two daughter cells. At the same time, they can also reproduce by producing a cyst. This cyst will actually be formed when the environment is not favorable, and ultimately the cyst will germinate again to produce normal trophozoite whenever there is favorable condition. Sometimes this cyst will actually produce a large number of cells, so it is also called multiple sporulation, and this particular structure normally will increase their number in nature and thereby help them to sustain the environmental condition. Another way that they reproduce is conjugation. As you can see here, the paramecium, they normally reproduce by conjugation. The two paramecium will come close to each other. They will develop a point of contact through which the nuclei or nuclear material can be exchanged, and thereby they will increase in their vigor. Now, majority of protozoa are causing infection, but some of them are actually helping in the degradation of complex substrate. So they are also taking part in bioremediation. Helminthology is the study of helminth. Now there is a controversy that whether helminths belong to the group of microbes because of their macroscopic size. But some of the helminths are microscopic in nature, and broadly they are classified as tapeworm and roundworm and the flukes or the liver fluke. And as you can see that this is a typical liver fluke which is having a typical flattened body with anterior and posterior sucker causing infection in cattle. This is a typical nematode. Now nematode, they can be parasitic in nature, causing disease to human, like the roundworm causing ascariasis, also can infect plant and causing different nematode induced diseases. There will be another group of organism which is commonly the tapeworm, which is causing infection to the human intestine. They are causing mixed infection in human intestine. But though they are present in the human intestine, they have their own niche or the area from which they draw nourishment. That's why on a regular basis we should take an anthelmintic drug and keep our intestine free from this helminth attack. Now usually these drugs belong to some specific chemical compounds which normally degrade the helminth and make our body free from helminth attack.
Sir, I would like to ask a question.
Yes.
Can parasitic infections be treated using vaccines?
Yeah, you see, it cannot be treated. Like you see, if there is a tapeworm, the tapeworm will survive in your intestine with the help of sucker. So any type of prevention like a vaccine will not prevent its attack. We have learned the different scope of microbiology, the domain of microbiology and the different microbes. The different groups include the virus, the bacteria, the algae, the fungi, the protozoa and the helminth. And also we have studied their cell structure, their reproductive pattern, their peculiarity of their life cycle and their economic importance, both useful and harmful. And this gives an idea that they are important in their own way. Maybe in future when we go to the next deliberation, we will try to see what are their different habitat and what role they play in their habitat.